Hello friends, welcome to a new lecture today. Now I would like to discuss about HIV. What is HIV? HIV is human immunodeficiency virus. So human immunodeficiency virus is the causative organism for AIDS, which is acquired immunodeficiency virus. This HIV can occur in two forms, HIV type 1 and HIV type 2. Okay, so what is the morphology of this HIV virus? So morphology of this HIV virus is, it is a spherical virus, number one. It is a spherical virus and it has been encapsulated. Okay, so this is the capsule or envelope. Okay, not capsule, I'm sorry, envelope. It is enveloped. So this is the envelope. So this is the HIV virus. It is the envelope. Okay, it is an envelope virus. It is a spherical virus. And it has two identical copies of genetic material. So it has two identical copies of genetic material. So these are RNA genome. Okay, so in an association with the RNA, there is an enzyme which is present in the HIV virus. So this is the enzyme. Along with the RNA genome, there is an enzyme, and this enzyme is called as reverse transcriptase. Okay, reverse transcriptase. This viral core, which contains RNA genome and reverse transcriptase, this is surrounded by a nucleocapsid. It is surrounded by a nucleocapsid. So this is the nucleocapsid. Okay. So this is nucleocapsid. This nucleocapsid has proteins. Okay, this nucleocapsid is composed of proteins. Now, the virus also contains a lipoprotein envelope. See this nucleocapsid? It is the nucleocapsid has two shells. Remember that the nucleocapsid which is there, that has two shells. The shell is, this is the inner shell, and there is also one more shell which is the outer shell. So this is the outer shell of nucleocapsid. Now, the virus also has a lipoprotein envelope. I have already shown you the envelope. This is the envelope, and this is the lipoprotein envelope. It contains two things. This envelope which I have shown, this has two things. One, it has lipid and also glycoprotein. Okay, that is the reason why we call it as lipoprotein. That is, it contains lipid and also glycoprotein. So we call it as lipoprotein. Now, the lipid which is formed, it is from the host. It is mainly derived from the host cell membrane. Whereas the glycoprotein part, it is from the virus. Virus will code this glycoprotein part. So this lipid part is from the host cell membrane, whereas the glycoprotein part is from the virus. So the major virus which is there, within the glycoprotein part, there are some projecting spikes which are seen. So there are some projecting spikes. I'll draw these projecting spikes with other color. Okay, so there are some projecting spikes from these envelope. So this is the envelope. So from the envelope there are some projecting spikes. Okay, so these projecting spikes, what does these do? These projecting spikes, these will bind to the host cell receptors. So these projections, they will bind to CD4 receptors of T cells. Okay, or any host cells. Okay, let them be any host cells. So that is the main use of this spikes. Now, what are the viral genes and what are the viral antigens? So this is the morphology. Now let us learn about the viral genes and their antigens. So what is viral genes? Viral genome, it contains mainly 3 genes. So these genes include, mainly there are, viral gene has two things. One, it has 3 genes which are structural. that is 3 structural genes, and it has 5 non-structural genes. It has 3 structural genes and 5 non-structural genes. These 3 structural genes are Gag, Pol, and Env. The 3 main structural genes are Gag, Pol, and Env. The 5 non-structural genes are, there are 5, right? Yeah, 5. They are Tat, Rev, Nef, Vif, and VPR. So these are 5 non-structural genes. So let us learn about each of them. Okay, in detail. So first gene is Gag gene. So this Gag gene, it will code for the core and shell proteins. This will code for core and shell proteins for the virus. So it is expressed as a precursor protein. First it is expressed as precursor. It is expressed as precursor protein, which is called as P55. Okay, I'll write it in my next page because it's a little bigger one. Okay, this Gag. So this will, Gag. This will express as precursor protein, which is called as P55. This precursor protein, it is cleaved, or it is divided into 3 proteins, which are P15, P18, and P24. This P24 antigen, which is there, this is the major core antigen, and this major core antigen, it is detected in serum during early stages of infection. During early stages of infection, it is seen, early stages of infection. Okay, and this will decline after appearance of antibodies. Our body will produce antibodies. Once the antibodies appear, so these will decline. So if you see, the virus has entered here. Within a few days, the P55 antigen will appear, it will peak, and now the antibodies which are there, P54 antibodies, these have started appearing. So once the P54, so this is, yeah, once the P54 antigen starts to appear, okay, these will peak, and the P24 antigen will disappear. Okay, so this is what happens. So this is P24 antigen, this is P24 antibody. Okay, so this is what happens in about P55, that is Gag gene. Now this P18 which is there, this is called as shell antigen, whereas the core antigens can be P24, which is the major core antigen, or P15 and P55 are also core antigens. This is about the Gag gene. Now let us learn about the next gene, which is Env gene. Okay, this Env gene, this will code for, as the name says, Env, so envelope. In the envelope there is glycoprotein 160. Okay, it will code for glycoprotein 160. Okay, so this glycoprotein 160, it will cleave into 2 components, and it will cleave into 2 components. One is glycoprotein 120 and glycoprotein 41. This glycoprotein 120, it forms surface spikes, whereas glycoprotein 41, it will form transmembrane pedicle protein. Okay, glycoprotein 120, it forms surface spikes, whereas glycoprotein 41, it forms transmembrane pedicle protein. This glycoprotein 120 which is there, so this is the major envelope antigen. Okay, now the antibodies to glycoprotein 120. These will appear after HIV infection. They first appear after HIV infection, and they are present till the terminal stages of infection. Till the terminal stages of infection they will present. So I'll just show you. Yeah, if this antibody, so this is the graph. These antibodies to glycoprotein GP, they are the first to appear in HIV after HIV infection, and they will be present till the terminal stages. Here there is infection. Okay, so this is antibody to glycoprotein 120. Okay, now let us learn about the next gene. The third structural gene is called as Pol gene. Next structural gene is Pol gene. This Pol gene, it codes for reverse transcriptase enzyme. It codes for reverse transcriptase and other enzymes. So this is expressed as a precursor protein, which is P100. And this P100, it cleaves into 3 proteins now. It will cleave into 3 proteins. One is P31, P51 and P64. All these are required for reverse transcriptase production. Now these are the structural proteins. Now let us learn about the non-structural genes. Non-structural genes. So these non-structural genes, they are present in all retroviruses. So these non-structural genes, these are the structural genes which are there, which I have discussed, like Gag gene, Pol gene, Env gene, these are present in all retroviruses. Whereas these non-structural genes, they are specific to HIV. Okay, so the first structural gene is Tact gene. This Tact is Transactivation Gene. This is Transactivation Gene. Okay, what does this Transactivation Gene do? This is mainly for a factor or a protein, which this will code for transactivating factor. It will code for a protein called transactivating factor. This transactivating factor will increase viral protein synthesis. This will increase viral protein synthesis. This transactivating factor will cause viral protein synthesis. Okay. So this is Tact gene. The second gene is Rev gene, or it is also called as regulatory of virus. So this Rev gene, it is required for expression of structural genes. So we have seen that there are 3 structural genes, right? Like Gag gene, Env gene, Pol gene. So this is required for expression of structural genes. Okay, so this is Rev gene or regulatory gene. So this is Rev gene, it is required for expression of structural genes. The third gene, which is Nef gene. Nef gene is also called as negative factor. So this negative factor, it is responsible for regulation of latent stage. It regulates latent stage of the vaccine. As I have said, it is negative factor, so it will help to regulate the latent stage of virus. Okay, now the third gene, non-structural gene, the fourth gene, non-structural gene, is which is important, is Viv gene. Viv gene is viral infectivity factor. Okay, gene. This viral infectivity factor gene, this will, as the name says, help in viral infection, or it will help or increase the infectivity of the virus, so that the virus can easily infect the human beings. Then the fifth gene, which is important, is VPR gene. This will activate the promoter region of the viruses. This will activate the promoter region of virus. The next gene which is important is VPU gene or VPX gene. So if you see, This VPU gene, it is seen in HIV-1, whereas VPX gene, it is seen in HIV-2. But both these will activate, or these will code. First, these will code for small viral proteins. The function of these small viral proteins is unknown. So these proteins have unknown function. And now these will also activate maturation and also release of viral progeny. Okay, so this is two types of gene, VPU gene and VPX gene. Then there is also one more sequences which are called as long terminal repeat sequences. These long terminal repeat sequences, these are present one at either end. Okay, they are present at one at either end, and these give promoter, enhancer and even integrator signals, integration signals. So these are the different structural and non-structural genes which we see in HIV. See guys, these genes, it is really difficult to remember them, but there is no way to remember them. You'll have to just try to expand it. It's simple. If you expand the genes, you'll easily remember it. It's Tact gene. So transactivation. See, you'll see T-A-T. So transactivation gene. So it will help in activation, transactivation, that is increased cell viral synthesis. It's Rev gene. Rev is regulatory gene. Regulatory of virus, right? It will help in expression of structural genes. In such way, it will regulate the virus. Nef genes, which is negative factor gene. What is negative factor in virus? In latent stage. In latent stage, it can't work. So even in that latent stage, it will have to work, and it regulates the latent stage. What is the next gene? I'm sorry, where is it? Yeah. The next gene is VIF gene, which is viral infectivity factor gene. So the name has infectivity. So this will activate infectivity of the virus. VPR gene, viral promoter gene, see, promoter region of virus. So that is VPR gene. So as a result, it will activate promoter gene region. VPU and VPX you'll have to remember, because they are the genes of unknown. They will produce some proteins which have unknown function. Then there is long terminal sequence. So you'll have to remember these. The only way that I remember is through the words. So thank you guys for watching my lecture. Thank you.